Hi everyone, my name is Swapna for the Royal Canadian Institute for Science, and today we have a really exciting opportunity to speak with Dr. Brett Finlay, who's come here all the way from the University of British Columbia, and he's the author of Let Them Eat Dirt. He's a world-renowned microbiologist, and we cannot wait to chat with him. Today we have with us Dr. Brett Finlay, who is a Peter Wall Distinguished Professor in the Michael Smith Laboratories at the University of British Columbia. Thanks for joining us, Dr.
Pleasure.
Finlay.
Yep.
Today, maybe let's start with how you came to be in microbiology. When did you know, and how did you know you'd caught the research bug?
Well, like most scientists, it's a bit of a circuitous pathway the way that you end up. So both my parents were scientists. Mom was a botanist, dad was a zoologist, ornithologist type thing. So I grew up in nature, looking at bird nests and plants and things. I really liked it. I thought I'd be a paleontologist, dinosaur bones. So I scrubbed dinosaur bones in high school and things. That was kind of cool. And then trying to figure out what I wanted to do in science, I really enjoyed chemistry and I really enjoyed biology. So I went to the chemistry department and said, here's the curriculum. I said, well, where's the biology course? I said, we don't have biology, you're a chemist, right? And then go to the biology department. Where's the chemistry? I don't need any of that, you're a biologist, right? So then I was just thumbing through the syllabus in the old days when they actually had hard copies of what departments do, and there's this thing called biochemistry. And I had no idea what it was, but it's in biology and chemistry. So sign up for it. So I signed up for biochemistry. And you actually didn't take a biochemistry course until your second year. But I loved it because that's basically the chemistry of life. Mm-hmm. And then in terms of, so then I was studying these things, and then I realized that you need research experience, right? So our neighbor, he was a neurologist, neurobiologist type thing, and he said, oh, you should come work in my lab for the summer. We just put these axons from squid down in the dish and put P theta 2 in one end, and we watch it go like this. I said, well, that sounds interesting. And then I happened to talk to my biochemistry prof. I said, well, that's not real science. You should come work with me. I work on these surface molecules coming out of bacteria. They're called pili or fimbriae. And these things allow the bacteria to adhere to things, and they also transfer DNA through this into other bacteria. And so in my third year of undergrad, I started working in that lab. And I really enjoyed that. I decided I eventually did my PhD in that lab on these structures. And then for a postdoc, the decision came, and this is right when molecular biology was moving into eukaryotes, mammalian cells, because it was in microbes until then. And everyone was jumping ship. I said, well, I like these microbes so much. So I decided I really wanted to study how microbes interact with mammalian cells, because then I get the best of both worlds. I get the microbes and these cells. Right. So I went to Stanford for my postdoc, where I actually then did some pioneering work on how microbes, actually certain microbes that cause disease, get into mammalian cells. We call this invasion. Mm-hmm. And so that started my work on Salmonella coli. And so we basically did, that's pretty much my career the last 30 years, was on studying how Salmonella, pathogenic E. coli, cause disease and what molecules actually do this. And then about a decade ago or so, we actually have lab retreats every year, and we go away and think about not what you've done, but what are you going to do type things. And we drink a lot and have these free-for-all discussions about, well, where might we go? And the discussion came up, what happens to your gut is filled with all these bugs. So if an E. coli or Salmonella, you go into the gut, there's all these bugs, what do you do? And with all these other things, and no one knew. So we sobered up, looked at literature, and bottom line is there's very little known on it. When you get cholera, you basically have a monoculture of Vibrio cholerae. But other diarrheas, E. coli, Salmonella, no one knew. So that started us into the world what was then called normal flora, which we now know as the microbiome, and we did some really interesting studies there. And two things really convinced me was important, because we could, if you take a mouse strain that would always die of disease and you did a fecal transfer from a mouse strain that didn't die of disease, you could actually save this mouse, and vice versa. You could change a lethal or non-lethal disease just by doing a swap of feces, which says it's very important. And then we worked on an organism called Salmonella typhimurium. It causes, so basically it causes typhoid fever in mice. That's where it gets this name from. But if you or I get it, we have gastroenteritis, we have diarrhea. It doesn't cause typhoid fever in us. But what we found is, so we took these mice, we treated with antibiotics. Normally it causes a typhoid fever in mice. It basically then causes a gastroenteritis, a diarrhea. So we completely shifted the disease in this animal just by treating with antibiotics. So those two things said these microbes are really important in affecting disease, and that led to our last 10 years or so in the whole microbiome world.
Incredible.
So there's my career in a nutshell.
in a few words. You mentioned that you were really interested in paleontology early on, and I've read that you were interested in anthropology. Has that interest, those roots, has that influenced your work in the microbiome, the behavior of people?
Yeah,
who passed?
it's a fascinating area. So, a few years ago, Canadian Institutes for Advanced Research was having calls for applications, and I wanted to put in a microbiome group of some kind because it was a really neat area. But we thought about it for a while, and there's lots of microbiome groups being formed, and so then we, ironically, I had some colleagues that were anthropologists that started talking about this. We got into exactly that. So in Brazil, they have a very high C-section rate, like 90%.
Right.
And we know C-section rates increase obesity and asthma and diabetes and things. So what's that going to do as a society? Or if a society changes some kind of food nutrition guidelines, which is going to shift microbes. Or when you're born, you inherit your mother's microbes as well as your mother's DNA. And so is that actually part of your evolution? And so we got into these really amazing discussions. Long story short, we basically formed this group. It's called Humans and Microbes, the effect of microbiome on human development, so embryonic development and things, human evolution, and anthropology, which is society and these things. And it's led to these absolutely fabulous discussions about really neat things. We're doing some very interesting stuff on, the question came, what happens when we colonized Africa and pre and post-European contact to the microbiome? And it turns out we have an ancient DNA guy, and you can actually take the calculus from teeth, take it out, and then sequence microbes. So we've now been collecting skulls from museums around the world, taking teeth out, scraping the calculus out, put it back, put the skull back in the museum, and we can actually sequence these microbes. And so we're looking at what happens pre and post-European contact and antibiotics and vaccines and things.
Wow, so
we're also sequencing Roman latrines and other things. So another really neat story is in we're looking at Italian graveyards back to the 4th century. You can dig these skulls up and do this, and they have the church records of what they died of. So we look at when pandemic plague comes through and people start dying of plague, we can see are there shifts in microbes allow people to increase their survival rate or something, because you can't genetically evolve that quickly, sort of thing. So it leads to some really neat questions all about culture and things. And so yeah, I mean, it makes you take a whole different look at how this all works, out of the molecular details of the nitty gritty, how it works. Yeah.
Oh, that's a really fascinating intersection of culture of one type and the culture of another.
Absolutely, yeah.
You've done a great deal of incredible work on E. coli, and I'm wondering if you can take us through a little bit of that journey.
Yeah, so when one says E. coli, to most people, there's two reactions. One, to scientists, that's the strain we use in the lab to clone everything. That's the workhorse of molecular biology and molecular genetics things. Then there's the other, the E. coli 157 and the hamburger disease and diarrhea and these other nasty type infections. So E. coli is a whole gambit of microbes. They all have sort of a central 80% conserved core, and they have what I call genetic upgrades that cause it to cause urinary tract infections or sepsis or hamburger disease. So our lab had studied diarrhea for a long time, and what we studied E. coli that cause basically severe diarrheas, and E. coli 157 also causes like kidney disease, something called hemolytic uremic syndrome. And we really want to figure out how does it work. So we basically go in and you can mutate microbial genes and things. And E. coli is fascinating, at least these E. coli are the diarrhea ones. When they bind to a cell, they bind really tightly, and they do an amazing thing. They basically rise up and they make the cell build a pedestal underneath it. So you get the E. coli sitting up on top of this nice pedestal on the cell. It doesn't go into the cell, it hangs out on top. So what our lab has done over the years is sort of figure out how it does that. And it's very sophisticated. What it does is it comes in and actually it's got this specialized syringe system that injects a bunch of bacterial proteins into the mammalian cell. And these proteins have different jobs once they're inside the cell to affect the immune response and the cytoskeleton things. And a lot of them affect the cytoskeleton, the actin, which then builds the pedestal these bacteria on. But the most neatest finding we ever made was that it actually injects a molecule into the host membrane that anchors there, and then the bacteria then locks down onto that. So it injects its own receptor into the cell. So it's kind of like if you're climbing, you pound in a piton and hang on to it, right? And this is unprecedented in the microbial world. But that's a key protein for E. coli. So once we knew that, we thought, well, how can we stop this? And when you stop the disease, and the way you get E. coli 157 is cows carry it, but they don't get sick. So you get it two ways. Either you get poorly cooked ground beef, That's why it's called hamburger disease. So
Right.
always cook your hamburgers well done.
Certainly.
You know, never rare, no rare hamburger. Or you get it because the cows carry it and they defecate, it goes in the environment and it gets in the water. And that's where the Walkerton outbreak came from in Ontario a few years ago. And also the water is then used to water spinach. Like right now there's a big romaine lettuce outbreak going on in the States, probably because the water they're fertilizing has this cow contaminants. So, you know, as science goes, I was out, so what do we do about this? We know this is cool, it's key virulence factor, what can we do? I was out running in the woods in the downland lands of UBC, and you know, your mind's just on autopilot. And I was thinking of a kid vaccine, because the kids are the ones that really get this horrible disease. Could we vaccinate the kids for this? And it's just like one of those silly ice cream in the forehead moments when I realized, it almost caused me to stumble. Well, let's vaccinate the cows, because they carry this thing, and then it's the humans that get sick. So we did some really neat experiments with some people in Saskatchewan and cow lots and things that proved we can actually vaccinate the cows for this. It works beautifully. We can actually prevent the disease in cows. So the question is, why aren't cows vaccinated these days? The science is all there. But that's when you get into policy issues, and farmers say, well, I'm not going to pay for the vaccine because my cows don't get sick. You know? And silly things like, is this an agricultural product or a food product? You're immunizing animals to prevent a human disease. So where does that fall in the government kind of thing? And it's just bottom line is it's not gone forward because of all these, you know, all these other issues. They're non-science issues. We know that if you vaccinate every cow in Canada, you basically save $200 million a year in healthcare costs of this thing. But you know, no one wants to spend that much money to vaccinate the cows. You know, prevention versus outbreak. So every time there's an outbreak, and we had that XL outbreak in Alberta, I was like, vaccinate the cows. We have this technology, but it hasn't gone in. So that's been a big frustration that we scientifically prove this works beautifully, but it's a whole other story of getting society to then use it.
Yeah, so that's the next step, trying to figure out how to get it actually implemented.
Well, this whole science policy thing is a big deal, and I assumed once you have the science, it's easy, you just get it done, right? And I didn't realize that's actually just the first step of a very long and arduous process.
So we're learning more and more about the extent of the influence of microbes, and we as humans are made up of more microbes than we are of human cells. I'm wondering, I think I've heard you speak about this before, the effect of microbes on our IQ or intelligence. Can you speak to that?
Yeah, so first of all, there's at least as many microbial cells in and on us as human cells. And they encode 150 times more genes than there are human genes in us. So that's really something when you start thinking about all sorts of things from personalized medicine, adverse drug reactions to all these. We know they have major effects on us. And everything from how the brain develops to the gut develops, the immune system develops. And in terms of IQ bugs, we're getting into a speculative area. So there's this thing called the gut-brain axis.
Yes.
So everyone knows that the brain controls the gut, right? But what we're now realizing is that a lot of the microbes in the gut have effects on the brain. And for example, if you take an anxious, a stressed, or depressed animal to fecal transfer to a normal animal, you can make that normal animal anxious, stressed, or depressed just by moving the feces over. There's lots of correlations with autism, and that their gut microbes are different. And more recently, we're finding some very fascinating stuff in Parkinson's and Alzheimer's being microbial linked. The whole intelligent question, we're not there yet. So we're doing experiments. We're now looking at young kids and their ability to learn music and also speech, and we're characterizing their microbes. We know that if an animal is raised germ-free with no microbes in it, their brain doesn't develop normally. But whether there's IQ bugs and things, that's yet to come. But I'm sure that the microbes affect how the brain develops.
Yeah. Speaking of the gut-brain axis, I think there's been some work done on IBS, irritable bowel syndrome.
Right. Yeah, it's the depression associated with irritable bowel syndrome. I mean, clinical irritable bowel syndrome, ironically, it's not an irritated bowel. You don't see clinical inflammation in the gut, and I think it's a brain type thing. And there are some probiotics, for example, that work in the brain that affecting IBS type things. So yeah, I think they influence it.
Yeah. I was wondering, so athletes who travel a lot for playing away games or tournaments or whatnot will be, I suppose, exposed to a great deal of different environments.
Yeah,
and
stress.
Different, and also therefore different microbes. So I wonder if that could be an area for focus in figuring out how to maybe maximize or optimize performance through microbes.
Yeah, so in this new book, there's a whole thing on exercise. Microbes really affect exercise for sure. In terms of traveling athletes, microbes affect the circadian rhythm. There's a lot of data on that, so jet lag and things. So, and then we also know they affect exercise, long-term survival, and their ability under stressful conditions and things in lab animals. So I wrote a sort of a tongue-in-cheek section. What's the ideal bug doping? So how could you design a cocktail of microbes that do this and this? So one of the things is exercise, you get a lot of inflammation, right? So there's microbes that decrease inflammation, for example. You put some of those in there. You could get microbes that, affect sleep and circadian rhythm and things. So it's a bit of tongue-in-cheek, but how you can do bug doping instead of drug doping, and no one could test you for that because there's no test for bug doping, right? So it's not that, we're still in the early days of figuring out which microbes do exactly what, but I think, there's microbes that affect bone density, for example, which is a big thing in athletes and things, and sarcopenia and stuff. So yeah, I think we're just really starting to get into the whole exercise concept of it, but there's studies that, on a professional rugby team versus controlled people, there's very difference in the microbes, and it seems to be affecting how they haven't done the experiment. You take a superstar athlete, Michael Phelps or whatever, and do a fecal transfer into someone else. They turn into Michael Phelps. I think there's a little bit of genetics involved in that too. But it is an interesting concept. Could you do enhanced performance from these microbes? Like people that run the Boston Marathon, as soon as they finish the Boston Marathon, their lactic acid metabolizing microbes are big in number because as you exercise you make lactic acid, and that's what causes pain in muscles, so you break it down, right? So if you had more of these things that could metabolize lactic acid, you could basically theoretically run longer and faster and things. So maybe you bug dope with the lactic acid metabolizing things before the marathon, you go faster. The experiment's not being done, but the theory is there now. So maybe
new doping possibilities.
Absolutely, bug doping, I love it.
The next question I really wanted to ask you is, what role do you see microbes playing in the application of the future of personalized medicine?
I think it play a huge role. So you and I are 99.9% genetically identical, minus a chromosome, but people are really similar genetically. There's this massive movement, well, let's personalize medicine. And yeah, we're slightly different with a few little snips and things, but really we're very similar genetically. Microbially, we're completely different. Everyone's got a unique set of microbes. At best, you share maybe 50% of the microbes with the person you live with, and there is no one microbe that's conserved in everyone. So everyone's different. So if you take a drug, who breaks the drug down? You don't, the microbes do. Yeah. Who forms these secondary metabolites, may or may not have adverse drug reactions or make sure the drug works. And there's some fascinating examples coming out about how microbes really influence drug activity. For example, digoxin, which is a heart drug, it turns out that there's certain microbes will break it down or not. And if you have these microbes that break it down, it doesn't work in you. Yet it'll work in me because I have the right microbes, right? So they thought, well, maybe we could give you certain microbes and then digoxin, so it'll work, for example. And we're finding this, for example, there's a fascinating new, wonderful area of cancer chemotherapy called immune checkpoint inhibitors. And this is where you basically use antibodies to relieve repression of the T-cell immune systems, and then these T-cells can attack tumors. And it works really well for hard-to-treat tumors like malignant melanoma and small lung carcinomas, except it only works in 20% of people. Well, 2 years ago, researchers showed that in mice anyways, in microbial dependent, and last month there were 2 fabulous stories that came out showing that depending on your microbial composition, we can predict whether those drugs are going to work in you depending what your microbes have. Wow. So you can see how you can start to get personalized medicine, right? And there's some fascinating studies in diet that you can actually, there's now a company that will actually predict what foods you eat based on your microbes, whether they cause a glucose spike, which then leads to obesity and diabetes. And if you follow the diet guidelines based on your microbial composition, you have a personalized diet. It's just annihilating the standard diets that everyone uses and they never work, right? Yeah. So I think it's going to play a major role, and I always tease the personalized medicine people that they're working on the wrong things. You're working on just a small percentage of the genes that are actually active in us. And so I think personalized medicine is going to have to embrace this huge complexity of microbes, because that's what's making us unique.
Yeah. That's really interesting to me, because I have a twin and I wonder how different we are really through our microbes.
Yeah, there was just a study coming out that genetics plays, they're estimating between a 2 and 10% composition in terms of the microbes, the environments, the rest.
Mm-hmm.
And there's lots of studies showing that twins living on the other side of the world, their microbes are completely different.
Right.
You know, it's when you're living together in the same house and eating the same things, yes, you're similar, but as soon as your environment changes, you change the microbes.
Mm-hmm. So she's gone to another province right now, and when she comes back, we should have.
You should do the
different
fecal
microbes.
analysis. It'd be really fun. Yeah, there you go. See how different you really are.
So you've also co-authored a fascinating book, Let Them Eat Here.
Thank you.
And I highly recommend everyone get that and have a look at it and read it through. I'm really wondering what you feel this can do for the public, and why is it important to engage the public in the work that you do?
Yeah, so I mean, first and foremost, I'm a scientist, and so I do what scientists do. We publish papers, we talk to other scientists, and that's great. But when you take a step back, why are we doing science? I mean, you want to improve the world, at least I do. Most scientists do. It's for the benefit. And ironically, we're actually paid by taxpayers to play, in a sense, if you can call it that. I mean, we use taxpayers' money to do this. And I think it's really important to return that knowledge to society however we can in terms that people can understand. And the really neat thing about the microbes is not too hard to explain. You can sit beside someone on a bus, you can tell them what a microbe is. They've heard of a few of them, quantum mechanics or, some crystalline structure of a sodium atom is difficult. So this is something people get, and the public has really embraced this whole area of microbes, fecal transfers and things. It's gross, but it's fascinating kind of thing. So we wrote this book because we really felt we were doing some studies in early life microbes and the effects on asthma. We had some really neat scientific findings that basically we can take a 3-month-old kid's feces and tell whether or not they're at risk for asthma based on the microbes.
Which is fascinating.
And then I was at a meeting where there's another person talking obesity, another person talking on gluten sensitivity, and they too came back to the same conclusion. It was really weird. It's always early in life because that's when these microbes really have their effect. So a postdoc, I wrote it with Claire Arietta. She was in my lab at the time doing the asthma work. We sort of said, well, we should write a book on this. And I'd never written a book before, but I felt very strongly about communicating science. So we did this. We're now working on a documentary on this, so people don't have time to read a book, and that's really exciting. And then I'm running another one now with my daughter, who's doing a PhD in gerontology and geography, affect the built environment of the elderly. So we're doing microbes and aging, and it's how to healthy age with your microbes. And this has just been a fascinating area. If you take the top 10 reasons why you're going to die, only one's obvious in microbes. That's pneumonia, number 8. And but 9 of those 10 now have microbial links, and Alzheimer's and cardiovascular disease and all these neat things. So. And the good thing about microbes is that you can change them. You can't change your genes yet, human genes, but we can change microbes really quickly. So there's a really lot of excitement in this area. So what I'm trying to do is convey the science as we know it today, because there's a lot of hype, fecal transfers, probiotics, prebiotics type thing, versus actually what do we really know scientifically, what's been proven in clinical double-blind placebo controlled trials to actually work. Fecal transfers work for C. difficile. In these clinical trials showing they might work for ulcerative colitis, inflammatory bowel disease, but all the rest, there's no data. Yeah, it's coming.
Yeah.
So I'm a very strong proponent of, if we find this interesting stuff, I mean, we can help kids. I mean, I think if a mother changes the way they raise their kids, they can actually have an impact on the kid's effect for asthma, obesity, diabetes, long-term health period. So I feel very strongly about it, and any chance I get to promote it in that sense, I think it's really important. Because people want to learn. Yeah. People do want to know about this. Most people do care about their health, I could say, if you brush your teeth 3 times a day, you have a 50% decrease rate of dementia later in life. You can do that. And we know because it affects the microbes and gum health, which causes inflammation, which then triggers dementia and Alzheimer's type thing. So, yes, I've published lots of science papers, but ironically, I think this has gotten a lot more attention than any paper I've ever published in my life, and I'm glad about that because it's a great way of getting science out there for the people.
It's a fantastic way of getting science out to the people, and it's very important. As you say, you can really change your microbiome, so this is something that people should know about.
Yeah, I think so, yeah.
Right. Well, thank you very much for joining us. Really
that was good
appreciate
fun,
your time.
Thanks.